ক্যান্ডিডাইন ব্যাল ফ্লুইড নো করতে থাকে চলছে। ঠিক আছে, বসো তো।
স্যার আপনি সামনে চলে আসুন।
না, আমি এখানেই থাকতে হবে।
স্যার আপনাকে দেখতে পাবেন না ভিডিওতে।
না, ভিডিওতে দেখতে হবে না। বসো বসো বসো। আচ্ছা, so today we will discuss two separate topics. One is gram stain and another is motility. বাংলায় সবাই বাংলা? ঠিক আছে। আচ্ছা, তাহলে আজকে আমরা দুটো টপিক ডিসকাস করবো, একটা হলো কি গ্রাম স্টেন আর একটা হলো কি মোটিলিটি। ঠিক আছে। আচ্ছা, তা গ্রাম স্টেন করার আগে আমাদের যেটা জানতে হবে সেটা হলো কি how to properly make a smear. So what is a smear? A smear is a uniform film of material made on a glass slide or a cover slip. ঠিক আছে। So the point to be noted here that the definition contains both the glass slide and cover slip. So you have to know the dimension of glass slide and cover slip. So the dimension of glass slide is around 7.5 cm into 2.5 cm, and cover slip is usually 2 cm into 2 cm or 2.2 cm into 2.2 cm. Now we have to before making the smear we have to properly clean and label the slide. For cleaning the slide, it can be done either physically or chemically. So for physically it can be done by wiping and flaming. Okay, so first you have to wipe the slide with a clean dry cotton cloth, then hold it with a forceps in the blue flame of the Bunsen burner and for few seconds, or you may just pass the slide 6 to 12 times to make it grease free. You are just roasting the grease which is on the slide. Or another option is there in the physical method. You can moist your fingertip, just rub the slide with your fingertip, then the moist fingertip has to be touched on a soap, then that fingertip has to be rubbed on the slide, and then you have to wipe the slide. That's also a way you can clean the slide. Or in special circumstances, the cleaning can be done chemically. For this, you have to take potassium dichromate in concentrated H2SO. You have to dip the slide in the potassium dichromate in H2SO4 solution for few days and at room temperature. You have to keep that at room temperature. Then you have to take out the slide and wipe it and use for smear making purpose. Now every slide must be clearly labeled. For labeling, you can either use diamond marking pencil or special marking pen. The ink of the pen should not be washed away during staining procedure. Okay. Now let's dive into making smears. For making smears of fluid material such as broth culture, specimen of urine, sputum, pus, other body fluid, one loopful or more is taken with the help of inoculating loop. This is the inoculating loop. This is the inoculating loop, and with the help of the inoculating loop, just take one loopful of the specimen and evenly spread it on the slide. Then the film is allowed to air dry and then heat fix and then used for staining. Next, for purulent or pus material, this can be also the smear can be done by inoculating loop. In case of sputum, you can use a clean stick to transfer the purulent and caseous part of the sputum on the slide, or if a nasopharyngeal swab has been sent, then the swab has to be moved in a rolling fashion like this, so that the Neisseria, it is a intracellular gram-negative diplococci, that Neisseria and the cell containing Neisseria is not lysed. So for any nasopharyngeal swab, you have to use this rolling type of motion for making the smear. So it avoids damage to the past cells. And in case of solid material, one loop full of normal saline is taken and placed on the glass slide, and inoculating loop is sterilized and minute quantity of culture is obtained, and then emulsion is made on the glass slide, and then it is air dried and then heat fixed and then stained. So for any fluid, especially let's say CSF or bronchoalveolar lavage, this contains very minute amount of microorganisms. So for those cases, this centrifuge machine comes into help, and we have to just centrifuge the sample, and from the deposit we have to make the slide. Okay. Next. So how a ideal smear looks like. A ideal smear is typically 3 cm into 2 cm. It is kind of oval in shape, and it is not too thin nor too thick. So how is an ideal smear? An ideal smear is so that if you watch a newspaper through the smear, you can read the letters.
Basically, it is transparent.
Transparent, but not that much transparent. So what we want a So we wanted unicellular layer. Unicellular thick layer for ideal smear. So these 3 points of ideal smear is very much important. Okay. Now fixation. We are going into the part of fixation. After preparing the smear, we have to fix it so that when it is washed during staining, the smear part does not get washed out. For fixation, we have to first air dry it thoroughly. Just keep it in the free air, it will dry. If we immediately start heat fixing the liquid on the smear, then it may cause boiling and precipitation of unnecessary material. So what we want first to air dry the smear, then on applying heat, the protein in the smear coagulates, and that actually fixes the smear with the slide, and the structure of the cell of both pus cell and bacterial cell are preserved. Okay. So now this fixation can be done either physically or chemically. If done physically, what we just told, that is the heat fixing. So overheating should be avoided, as it distorts the cell. Okay. So just put the slide a little bit above of the Bunsen flame, it will fix the slide. For chemical fixation, the main material used is alcohol. Alcohol can be either absolute alcohol or 70% alcohol. Absolute alcohol, that is absolute methanol and absolute ethanol. The leukocyte and pus cells are well preserved, hence recommended for fixing smears of gram-negative diplococci, which are intracellular in nature. Gram-negative diplococci, intracellular gram-negative diplococci, example is Neisseria gonorrhoeae. And the 70% concentration alcohol is usually used for Mycobacterium tuberculosis, because this 70% concentration rapidly kills the Mycobacterium tuberculosis in the sputum sample, so it is useful in that case. Other chemical fixative like potassium permanganate, formaldehyde vapor, formalin, mercury chloride, osmic acid, etc. also are used. this potassium permanganate is recommended for anthrax bacilli. The formaldehyde vapor is used mainly for mycobacterium species. Okay. Now we are going per se into the staining part. So this staining, there are different types of staining. Simple staining, then this is the simple staining, this is the differential staining, this is the special staining. So our point of discussion today is differential staining. Why it is called differential staining? Because two or more stains are used to differently stain bacterial types or bacterial structures. Okay. So examples are gram stain, acid fast stain, Giemsa stain, etc. So if you look carefully, this is the acid fast stain, this is the gram stain. This is also this is actually methylene blue stain. Methylene blue is a component of acid fast stain, that is the Gill-Nielsen stain. Okay. Now let's go into the gram staining. The gram staining is classifies bacteria into gram positive and gram negative based on the chemical and physical properties of the bacterial cell wall. So some bacteria have more mucopeptide and some bacteria have less mucopeptide in their cell wall. So gram stain actually differentially stains those two types of bacteria. It was discovered by Hans Christian Grams in 1982, which he published in 1984. Okay. So originally, 1882 and which he published in 1884. Okay. So the main, the original gram stain which he performed actually had aniline gentian violet as primary stain, lugolcidine as mordant, absolute alcohol as decolorizer, and Bismarck brown as counter stain. The original gram stain was later modified by many persons, namely Brooke, Kopelov and Barman, Jensen, Wizard and Peterson, Morel, etc. We will discuss them. Okay. Now, if we go into the gram staining procedure, step by step, if we look into the gram staining procedure, that we have to first use the primary stain, that is the crystal violet, for 30 seconds to 1 minute. Then we have to wash under free flow of water. Then we have to use the mordant. Mordant is gram iodine, that we have to keep for 1 to 2 minutes, then wash. Decolorizer is the most critical step, either alcohol or acetone alcohol, and this 5 to 15 seconds is depending on what we are using as decolorizer, and then we have to wash immediately. This is a rapid step and this is a very critical step, because if we keep the decolorizer for more time, then the slide will get over decolorized. Even the gram positive organism will also lose the primary stain, and if we do not decolorize, if we keep the decolorizer for less time, then it will cause under decolorization, and even some gram negative organism will lose gram positive. Okay, so after that we have to wash and use the counter stain. Usually what we use is saffron. Okay, now in place of crystal violet, other options what we can use are methyl violet, gentian violet, etc. And this, how this mordant or gram iodine work? This mordant or gram iodine actually forms a diiodine complex, crystal violet iodine complex. It is called CVI, crystal violet iodine, CVI complex. This CVI complex gets trapped in the cytoplasm of the gram positive bacteria which are hard to wash out. Okay, and how this decolorizer works, we will go into different theories of gram stain. We will discuss that in that place. Okay. Now, for counter staining, we can use saffronin, we can use carbol-fuscin, we can use neutral red. These are other options other than saffronin. Okay. So after the gram staining, we have to observe it with cedarwood oil in 100x, and the gram positive organism will look purple or violet, and gram negative organism will look pink or red. And what we have to take control. What is the purpose of this control? The purpose of the control is, have we done the staining procedure correctly? If the staining procedure we have done is correctly done, then the Staphylococcus aureus are known gram positive organism, gram positive cocci in cluster will look purple or violet, and a known Escherichia coli colony will look pink or red. So this control is very much important, that our procedure is properly performed or not. So what we have to remember in this case is, there are mainly 3 steps: this primary stain, this mordant step, and this counter stain step. We have to remember them by either 1, 2, 1, means 1 minute, 2 minute, 1 minute, or we can remember this as 1 minute, 1 minute, or 1 minute. Okay, both are correct. 1, 2, 1 is also correct. 1, 1, 1 is also correct. Okay, and there are some modifications that you can keep this in 30 seconds, 30 seconds, 30 seconds. This is also a acceptable rapid gram stain modification. Okay, we will discuss that later. And other modifications also there. So these are all acceptable modifications. Okay, and this decolorizer step, if we, let's say, doing it practically, then for this step, and this step, you can turn off the flow of the tap. But for decolorizer step, you have to keep the flow of the tap open and running tap. The tap should be running, right? The tap should be running, and you have to hold the slide in a tilted position and drop the decolorizer drop by drop, and within, you have to just count 1, 2, 3, 4, 5 and wash. Or when we count 1, 2, 3, 4, 5, then it can become rapid. So how to make it proper 5 seconds? You have to count 1001, 1002, 1003, 1004, 1005. That way your counting becomes almost equivalent to second. So you have to count 1001, 1002, 1003, 1004, 1005, and then you have to wash. Okay. Now let's go into the leading theories of gram stain reaction. So the main theory is cell wall permeability theory. So what is this theory? According to this theory, some bacteria have a thick mucopeptide layer and some bacteria have thin mucopeptide layer. The mucopeptide means that NAG and NAM connected by peptides. N-acetyl glucosamine or N-acetyl uramic acid connected by peptides and glycine bond. Yes, pentapeptide and pentaglycine bond. Okay. So the bacteria which have this thick mucopeptide layer are when stained by primary stain and the mordant is used, the diiodine complex is formed within the cell, and when the decolorizing agent is used, the diiodine complex cannot get pass out through the thick mucopeptide layer. But in case of gram negative cell wall, as the mucopeptide layer is thin, the diiodine complex easily get passed out from the cell, and the gram negative cell bacterial cell gets decolorized and it at the end takes the counter step. So this theory was proposed mainly by Saturn in 1963. Then comes the magnesium ribonucleate theory. So according to this theory, magnesium ribonucleate is responsible for gram positiveness and its absence is responsible for gram negativeness. According to this theory, there is a formation of insoluble complex of diiodine and magnesium ribonucleate in the gram positive bacteria. So actually this theory is telling that some bacteria contains magnesium ribonucleate, some bacteria does not contain magnesium ribonucleate. Those bacteria which contain magnesium ribonucleate forms that diiodine magnesium ribonucleate complex inside them and gets stained by the primary stain, and those who does not contain the magnesium ribonucleate gets stained by the counter stain. Okay. Now there is lipid content theory. This was proposed by Stern. Okay. This is a more recent theory. This theory says that gram positive bacteria only contains 4% lipid in their cell wall, while gram negative bacteria contains 20% lipid in their cell wall. So this high percentage of lipid in gram negative bacteria, when exposed to the decolorizer agent, mainly the acetone or alcohol, gets solubilized, actually gets emulsified and produce large pores in the cell wall of the gram negative bacteria, which actually facilitates the escape of the diiodine complex. But in case of gram positive bacteria, the lipid content is less and very small or negligible pores are formed when the decolorizer is added, that is the acetone or alcohol. So the diiodine complex cannot escape and the bacteria is stained by positive state. Now this acid protoplasm theory, according to this acid protoplasm theory, the gram positive bacteria retain primary stain more strongly because they have more acidic cytoplasm than gram negative bacteria, and that primary stain which is used in gram staining, the Jensen violet or crystal violet, is actually basic dye. So also the iodine, this makes the cytoplasm more acidic, which is used as a mordant, and this basic dye is, when entered into the acidic cytoplasm of the bacteria, it forms acid-base reaction and it gets stained. While the gram negative bacteria is basic in the cytoplasm of gram negative bacteria is basic in nature, hence there is no attraction between the basic cytoplasm and basic stain, so they are unable to retain the primary stain. Another theory is there which is called the isoelectric point theory. So what is the isoelectric point? Isoelectric point is the pH where ionization of the amphoteric compound is maximum. Amphoteric means containing both positive and negative charge. Okay. So isoelectric point range of gram negative organism is pH 2 to 3, while that of gram negative organism is pH 4 to 5. There are more unsaturated acid in gram positive organism than gram negative organism. This unsaturated acid gets oxidized during iodine treatment and more acidity is produced in the cytoplasm. Thus, gram negative organism, gram positive organism are able to retain the basic dyes at higher hydrogen concentration. So that is the isoelectric point theory. So next, limitations and factors affecting the gram stain. Gram stain, the result of gram stain may vary with the age of the culture, the pH of the medium, the viability of the microbes and the choice of stains. So if the gram positive microbes may appear gram negative and gram negative may appear gram positive if the culture becomes old. Okay, so this old culture, the culture older than 24 hours may contain a large number of dead, dying and degenerative cells which are physically and chemically altered So they cannot retain the gram positive state. So even though the organism is gram positive, they may appear gram negative. Now let's say the particular some organism when grown in a fermentable substance become acidic, forms acidic reaction in the growth, and if the staining is done from that medium, then the gram negative or gram positive nature of the organism may get distorted. Okay, so some bacterial gram positive bacteria like actinomyces, corynebacterium, mycobacterium, propionibacterium have cell wall which are particularly susceptible to breakage during cell division, resulting in gram negative staining of these cells. Okay, cultures of Bacillus, Clostridium are decrease in peptidoglycan thickness during cell growth causes some of them to appear gram negative. So a condition may arise that a single organism showing both gram negative and gram positive cell in a single slide. So those are termed as gram variable. Okay. Now let's say some bacteria which do not have cell wall, like mycoplasma, they are always gram negative. Some gram positive bacteria which are stripped of their cell wall are called L form. Though they are actually were gram positive, the L form will look the gram negative. Okay. Now let's say this mycobacterium, it is difficult to state, so it should be gram neutral, but actually we see that it is weakly gram positive. So this gram staining procedure, though is a very most widely used staining method in microbiology, but it also has some fallacies. So it should not be taken as the last word for diagnosis. Okay. Now application and modifications of gram. So this gram stain is usually the first step in bacterial identification and classification. Okay. So observation of the bacteria in clinical specimen provides a vital clue for diagnosis of the infectious disease and can help in estimation in the count of bacteria, and empirical antibiotic can be started on the basis of result of gram stain. Okay. Now the choice of culture media for inoculation can be made empirically based on the gram stain report. Let's say we have gram negative organism, we will choose Macon-Key agar. But if we have gram positive organism, then the Macon-Key agar only will not suffice. We have to add blood agar also. The blood agar will help grow the gram positive organism, and if there are some gram negative organism, that will grow in Macon-Key agar. Macon-Key agar has bile salt, that is the sodium taurocolate, that actually inhibits the growth of gram positive organisms. Okay. Now so rapid assessment of clinical specimen like CSF and urine. So when any CSF or acidic fluid comes into our laboratory, these are critical samples because these are sterile body fluid. Sterile body fluid, if infected by microorganism, is usually have devastating result, devastating outcome. So if we do rapid gram stain and we find that the CSF or acidic fluid has the presence of bacteria and pus cell, we must critically alert the clinician to start antibiotic on it is in different laboratory medicine there are different critical alert system, different critical and in our microbiology the sterile body fluid showing gram stain positivity is a critical alert. Okay. Now let's go into the common modifications of the gram stain. The gram stain was modified at different times by different person. The Brooks modification in 1922 originally was the modified the original Grams method by using 100% acetone or acetone ether mixture as decolorizer instead of absolute alcohol. Then Kopelov and Barman's modification in 1922. It is a modification of Brooks method. In this method, the staining solution used were methylene violet with sodium bicarbonate in distilled water as primary stain, and iodine dissolved in 4% NaOH solution as modern instead of alcohol, 100% acetone as a decolorizer, and basic question used as a counter stain. This method is sometimes modified by substituting acetone alcohol in place of pure acetone as decolorizer. A mixture of acetone and alcohol is recommended because it decolorizes more rapidly than 95% ethanol alone and is less likely to cause over decolorization of the smear than acetone without addition of alcohol. This method can be used in general practice. So, the acetone is a powerful decolorizer, alcohol is a less powerful decolorizer. So, what we are doing? We are mixing the both, and let's say 5% acetone and 95% alcohol, and we are using that for decolorization. If we use only 100% acetone, then we have to keep the decolorizer for 1 to 2 seconds maximum. And if we are using, let's say, only alcohol, then we have to keep the decolorizer for, let's say, 10 to 15 seconds. And if we are using acetone alcohol mixture, then 5 seconds is almost sufficient. Then let's say Jensen's modification. Jensen used methyl violet as primary stain, iodine and potassium iodide in water as modern, absolute alcohol as decolorizer, and neutral red as counter stain. Safranin or basic fustian were also available as an option, but not carbol fustian, as it stains gram-negative microbes so intensely that it may appear gram-positive. So, this can also happen, that if a gram-negative organism takes too much dye, then it may appear gram-positive. This we regularly face in our daily staining. Some gram-negative bacilli appear in dark color. Gram-negative bacilli should appear pink or red, but in sputum sample, we are seeing growth of, let's say, Klebsiella in the plate, and when we are corroborating with the direct smear staining, then we are finding that it is kind of dark colored bacilli we are finding. So, if someone is novice, then he may mistake that as a gram-positive bacilli. But actually that is not a gram-positive bacilli, that is actually a darkly colored gram-negative bacilli. So, because of that mucus of the Klebsiella, it tends to retain more gram-negative type. So, that may appear gram-positive. Okay. So, now let's say there is Wright's modification, Wizard's modification. Wizard's modification is what? These are mainly used for tissue section and used mainly for pneumocystis cyst. Pneumocystis zero cause pneumonia and other manifestation also. So this tissue section, which is usually done at pathology, the Wizard modification of gram staining is used for those tissue section. So in that Wizard modification, the modification uses carbol Jensen violet prepared from saturated alcoholic solution of Jensen violet and 5% phenol solution as primary stain, and gram iodine used as mordant, and aniline xylol is used as decolorizer. The counter stain is carmelam. Carmelam is carminic acid and potassium alum in water, and this is the Wizard's modification used in pathology for their gram staining purpose, mainly for finding the pneumocystis cyst. Okay, so there is Preston and Morell's modification published in 1962. The primary stain was used, that is ammonium oxalate crystal violet mixture, and the smear was washed in Lugol's iodine and further treated with iodine solution. The smear was decolorized using iodine acetone decolorizer and counter stained using dilute carbol fustian solution. This dilute carbol fustian, that is 1 in 10, is recommended for staining Vincent bacteria, Yersinia haemophilus. So this so many person has differently modified the gram stain. As we are microbiologists and you are the laboratory technologist, we have to know extensively about this modification. Otherwise, the MBBS person or any person from any other medical field need not to know this extensive modification. But as this is our domain, we have to know all this modification. Okay. So, and then there is a Brown and Braid's modification. This modification is used for staining Nocardia and Actinomyces species in the tissue. It is also helpful in staining fungi. This method uses 0.5% aqueous crystal violet or methyl violet solution as a primary stain and equal parts of acetone and 95% ethanol as decolorizer and 0.5% aqueous safranin or dilute carbolfustin as counter stain. So there are other modifications like Hacker's modification, then so these are all modifications. Okay, now let's go into the motility part. So this motility part we will wrap up rapidly. Okay, so the motility is ability of bacteria to move actively and purposefully through a liquid medium. So what is its importance? Two bacterial species, E. coli is motile, Klebsiella is non-motile. E. coli has fimbriae and Klebsiella doesn't have any fimbriae. So this can act as a differentiator point. Okay. Now the three primary methods of detection of motility is semi-solid agar stab method. It is a macroscopic method, wet mount microscopy, and the gold standard is hanging dog microscopy. Okay. So this semi-solid agar stab method, so this semi-solid agar, how this semi-solid agar is prepared? Depending on the type of agar, either Brazilian agar or New Zealand agar. If we are using Japanese agar. If we are using Japanese agar, then 0.4%, and if we are using New Zealand agar, then 0.2%. So if this is the agar concentration, then this agar is semi-solid in room temperature and it is filled in a 10 ml amount in a test tube and kept in vertical position. And then we have to take the test bacteria and with a straight inoculating wire, not a loop. We have to take the bacteria with a straight inoculating wire and we have to just stab the semi-solid agar up till 4 millimeter of away from the end of the tube. So there should be a distance around halfway. You can stab the straight wire. Okay, and there should be only a single stab, not multiple stab. Okay, so then you have to incubate at let's say the standard 37 degree and for examine at 6 hour interval, 1 day, 2 day and 6 day. We have to check the tube, and what we can see non-motile bacteria gives growth that only along the stab line. Non-motile bacteria shows growth only along the stab line and margins are sharp and defined and surrounding medium is clear, and the motile bacteria diffusely spread outwards towards the wall and foot of the test tube. Okay, so there are some cautionary pattern. What cautionary pattern? Let's say this finger-like outgrowth. Finger-like outgrowth means this was actually here. This is showing the finger-like outgrowth. Why this is a finger-like outgrowth? The stab was up to this level only. Then a crack was formed somehow in the agar. There was a crack in the agar, and the bacteria actually followed that crack only. Grown along the crack only. This is not the growth of the bacteria. This is actually a finger-like outgrowth, or some fan-like outgrowth may form. Why that fan-like outgrowth may form? Let's say I have inoculated a non-motile bacteria, and in that non-motile strain, what is the strain? Strain means, let's say I take sample from him and I grow a E. coli, and I take sample from you and I grow another E. coli. So your E. coli is, let's say, strain A and your E. coli is strain B. So what is usually seen that 98% E. coli, I am just let's say roughly 98% E. coli of all type of E. coli are usually motile, but 2% among them are usually non-motile. So let's say your E. coli is motile and your E. coli is non-motile. So I have confirmed that that your E. coli is non-motile. But that non-motile E. coli have to show this type of reaction, but some of that non-motile strain suddenly due to mutation becomes motile and show 1 to 2 shoots like this towards the, let's say towards the wall or towards the foot. That is a fan-like outgrowth from some point. So that is a motile variant of non-motile strain. Okay. So these are some cautionary patterns.
Sir, after step to state where the agar gel, you can put back or can put back, put out.
Yes, we have to just one step you have to make and put out the inoculating wire and burn it in the bones and sterilize, and you have to use again. Only one step is enough, nothing else. Okay, so there is two microscopic methods. One is wet mount, one is hanging drop. Wet mount is not much less important. How this wet mount is done? Wet mount is done. You have to just take a plain slide. You have to put a drop of the broth, or you have to put a drop of a normal saline. Then take a little bit of culture and emulsify the culture in the drop of the normal saline and put a cover slip. And then you have to watch under microscope. This wet mount is not a good method of viewing the motility, but the gold standard method of viewing motility is hanging drop. So this is the wet mount procedure. In this instant, let's discuss this, that how to actually watch this diffracting, this reducing light and defocusing condenser. Why is this important? This is important because there are true motility, there are Brownian movement, there are currents or drifts. So we have to differentiate true motility from Brownian movement and current or drift. Current or drift is when you are looking in the microscope, all of the particles are moving in the same direction. Then we have to understand that there is a current in the fluid, that fluid is passing to that direction. That is why all the particles in that field are going in the same direction. Okay, that is the current or drift. Okay, and let's say what is the Brownian movement? Brownian movement is jiggling movement, purposeless movement. But what is the most important? If you follow the movement, after some period you will find that actually there is no relative displacement of the particle. The particle is moving here and there only, but no actual movement to some other part of the field is not there. So that is Brownian movement. Okay, so that is the Brownian movement. And what is the true motility? True motility is purposeful movement and direction. It has direction. The Brownian movement is directionless, but purposeful movement in a specific direction, changing position, it is a true motility. And as we are seeing this in wet mount or hanging drop, these are unstained preparation. When we are trying to see this unstained preparation, the normal microscopic setting which we use for watching the bacteria is not useful. First, we have to focus. We have to keep the condenser and the focus and the objective lens, everything as we do in normal case. Then we have to defocus. We have to put the condenser to the lowermost position. This actually reduces the light, and the little bit of light that passes through the condenser actually gets reflected from those moving bacteria, and that helps us to watch the bacteria. So this defocusing the condenser in case of viewing a hanging drop method is an important thing, and we always watch the hanging drop method at high power, that is 40x. Okay, now let's actually detailed procedure of the hanging drop method. So we are using either this plasticine material, or if we are using a straight glass slide, normal glass slide, then we have to use this plasticine material. And if we are using this hollow carved special type of glass slide, then we don't need this plasticine material. Okay, and this is the actually the ideal thing. You have to use the petroleum jelly at the corners of the cover slip. Put a drop of water and emulsify a little bit of colony in the water. Then upon that cover slip, you put the inverted this special type of hollowed slide, and then you rapidly invert the whole slide. So that petroleum jelly covers creates a airlock type of thing, and where we have to focus? We have to focus at the air-water interface. Why we have to focus at the air-water interface? Due to the surface tension of the air, all the molecules that are larger than the water molecule will get concentrated at this air-water interface. That is the surface tension. That is due to the surface tension. And we have to focus in the 10x, that air-water interface, and then we have to change the objective lens to 40x. And in the 40x, after changing the 40x, we have to defocus the condenser. You have to put the condenser to the lowermost position. Then you will see different types of motility. Okay. So these are all written here. So why this petroleum jelly actually prevents dyeing and can be observed for long period. But actually, this if we are using plasticine, then maximum 1 to 2 hours that water drop is remaining. Otherwise, that water drop will vaporize. Okay. So next, let's go into different types of bacterial motility. So flagellar motility, which is the most common, the different type of flagellar arrangement. Atrichus means no flagella, monotrichus means single unipolar flagella, lophotrichus means a bunch of flagella at one pole, amphitrichus means one flagella or bunch of flagella at 2 poles, peritrichus means along the periphery whole thing has multiple flagella. Okay, so this monotrichus vibrio cholerae shows darting motility. This lophotrichus, tuft of flagella at one pole, it shows Helicobacter pylori. Amphitrichus, this Campylobacter jejuni. Peritrichus, the Escherichia coli, Salmonella species. This show tumbling and running motility. Okay. Now, this what is this gliding motility? Without flagella, with the help of pili. This is the gliding motility. This is the structure of a pili. This Mycobacterium, this Cytophaga. This show with the help of pili, they glide like worm. They glide like worm. This is actually this is the gliding motility. Okay. Now this twitching motility or jerky motility by type 4 pili is seen by this Pseudomonas aeruginosa and Neisseria gonorrhoeae. And another one is this swarming motility. Swarming motility actually is shown by formation of swarm cell. What is this swarm cell? Look, swarm cell. It is mainly shown by Proteus species, and Bacillus subtilis also shows that. And this is actually the figure of the picture of Proteus. This is actually the main colony, and look here, there is a margin. These are actually the swarming. What is swarming? Let's say bacteria is a unicellular organism. 10 to 25 bacteria come together, form a multicellular cell, multi-nucleate cell. That is a swarm cell. That swarm cell is highly active. Why this type of thing is seen in Proteus? Because they want more nutrition. So they go to different extent for finding that nutrition. So in a nutrition deficient media, they form that swarm cell, a multinucleate cell, 10 to 25 Proteus cell come together and form a swarm cell, and that swarm cell start swarming. Okay. Next, let's go to named motility style of different bacteria. There are some named motility style like this darting motility or shooting star motility shown by Vibrio cholerae, like shooting star. This motility will be like this. Okay, like this. Okay. Now tumbling and running motility. This tumbling and running motility is actually shown by Escherichia coli and Salmonella typhimurium. These are smooth directional movement, that is the run, and then interrupted by brief random reorientation, that is the tumble. Let me show you a picture. This is the picture. Okay. This is running, then tumbling. Running, tumbling. This is running, then tumbling. Okay. রানিং, তারপর আবার একটু টক্কর খেয়ে নিল, ডিগবাজি খেয়ে নিল, একটু দৌড়ালো, তারপর আবার ডিগবাজি খেয়ে নিল। হ্যাঁ, একটু দৌড়ালো, আবার একটু ডিগবাজি খেয়ে নিল। মাঝখানে ঠিক আছে, একটু দৌড়ালো, একটু ডিগবাজি খেয়ে নিল। ঠিক আছে, তো এইটা হলো কি টাম্বলিং। ঠিক আছে, আচ্ছা আচ্ছা, তারপরে হলো গিয়ে কর্কস্ক্রু মোটিলিটি। কর্কস্ক্রু মোটিলিটি ইজ শোন বাই দ্য ট্রেপোনেমা, দ্যাট ইজ দ্য মেনলি দ্য দিস ট্রেপোনেমা প্যালিডাম, দ্য পজিটিভ এজেন্ট অফ সিফিলিস, দ্য স্পাইরোকিক্স। The spirochetes are very thin and corkscrew like arrangement. This is the negative background negative background black dark down microscopy, dark down microscopy showing this spiral bacteria, thin long spiral bacteria, Treponema pallidum. These spirochetes show corkscrew motility, like this. Okay, corkscrew. Okay, now this Treponema pallidum also shows flexion extension motility. This flexion extension motility is this one. This one. It will go like this and then 90-degree flexion. This flexion extension motility is also shown by the Treponema. This flexion extension motility. Okay. Okay. So this falling leaf motility is shown by Giardia. Giardia is a hemoparasite. Giardia lamblia is a hemoparasite. Look here. Okay, this is Giardia lamblia. Okay, this falling leaf motility. Darting motility by Campylobacter and Vibrio, mainly Vibrio. Swarming motility by Proteus, Bacillus cereus. Tumbling motility and gliding motility by Mycoplasma. Tumbling motility by that E. coli or say Listeria. Corkscrew motility by Treponema. Okay, finally, thank you.
খাতা নিয়ে আসতে হবে তো।